we'll get started, so I don't want to steal any time. It's my pleasure to introduce Prashant Mali for today's seminar. I have to say, for quirks of the schedule, I actually haven't gotten to meet Prashant yet until right now, so I'm mostly going off his biographical information he gave. But reading through that, I'm very excited for this seminar. I think it's going to be a real treat. Prashant has done some extraordinary work, published in all the high-profile journals. He earned his undergraduate and master's degree both at the Electrical Engineering at the Indian Institute of Technology, Bombay, and then did his PhD at Johns Hopkins with Linzhao Cheng, studying the challenges of inducing and controlling human pluripotent cells. So I think based on that graduate work, he became very interested in the challenges of controlling these iPSCs and decided to go in a more technology direction, working with George Church, where he is tackling two major challenges. One is developing ways to cleanly and efficiently edit the genome of these cells, and then second, methods to differentiate iPSCs into transplantable tissue forms. So with that, please join me in welcoming Prashant.
Thanks for the kind introduction, and thanks to the department for giving me this wonderful opportunity to be here. It's really a privilege. And so for the purpose of my talk, I'm going to focus almost entirely on the work I was doing as a postdoctoral fellow in the Church Lab. In particular, I'm going to focus on a new sort of genome engineering tool set that has emerged pretty much over the past 2 years and really sort of taken over the genome engineering field, which is based on the CRISPR-Cas systems. And a lot of the foundational work in that field was basically done right here at Berkeley. So it's a special privilege to really come and be here and sort of present this body of work. So most of what I'm going to talk about is sort of my journey through the process of trying to develop this CRISPR-Cas genome engineering tool set. And a little bit towards the end, I'm going to diversify into the area of trying to use this system in a true causality sort of setting. So in terms of being able to engineer genomes or to be able to engineer DNA, really sort of the motivation comes from the fact that really the DNA is a sort of central material for life. Our genomes encode the rules for most life forms. They account for the vast diversity of flora and fauna that we see around us. And importantly, they also potentially underlie traits among certain individuals. And still more importantly, it's basically the aberrations in our genomes that account for a vast majority of the human diseases. So really, DNA is the fundamental material of life. And from the discovery that it is also the heritable form of genetic information to sort of the elucidation of the double helical structure, and finally, with the advent of Sanger and Gilbert sequencing, when we could finally reliably analyze this biomolecule, we've had a long march towards reading genomes. And in some sense, it culminated with the draft of the human genome about a decade ago. But since then, with the advent of next-generation sequencing, our ability to read genomes has really exploded, so much so that we're sequencing genomes at about $1,000 a genome. And as of 2011, over 250 eukaryotes and 4,000 bacteria and viruses have been successfully sequenced. So in some sense, our ability to read genomes has really grown very powerful, and we are very good at being able to read genomes. But in being able to interpret genomes, I think it is still a fair assumption to say that a decade removed from the Human Genome Project, much of how the genotype governs phenotype still remains to be explored. And we have powerful tools in that direction. For instance, since 2003, we have the ENCODE project that seeks to identify functional elements by correlating output with sequence data. And since 2005, we've had a large number of GWAS studies that seek to identify novel sequence variants associated with diseases or novel traits. But while these sort of powerful approaches help us sort of narrow down what are potentially the key causal genes in the human genome or other genomes, they do not really truly establish causality. And if you have to truly interpret genomes, we have to be able to establish causality. But to be able to do so, we really need to be able to achieve two major functions. The first of them is, if we wish to establish the causality of a particular DNA element, then we should be able to, in an ideal situation, we want to be able to perturb that particular DNA element, either by editing or regulation. And second, to be able to develop methods to assay the consequences of these genetic perturbations. So we really need tool sets on both the ability to perturb DNA and as well as to be able to assay the consequences of these genetic perturbations. And if there are facile approaches on both fronts, not only does it enable us to look at complex biological processes, but potentially to engineer life itself. For instance, we could start to impart novel functions in organisms. And most importantly, one can start to contemplate gene therapy in potentially not the too distant future. So for the purpose of my talk, I was just going to focus almost entirely on tools that I've been developing on the front of being able to perturb DNA, as well as being able to assay the consequences of these genetic perturbations. But before I kind of delve into sort of the details of these studies, it is sort of instructive to note that to be able to perform either of these functions, we need to be able to do this in a framework. And that framework is our ability to be able to recapitulate biological processes in a viable experimental setting. And in this regard, model organisms such as Drosophila and the mouse have been very powerful tools for biology. But I think in the context of human developmental biology, too, we're kind of entering into a very exciting phase. And that is really, in large parts, through sort of the advent of the human iPSC paradigm. For instance, now it is possible for us to take somatic cells from patients and convert them into iPSCs, and at least in theory, differentiate them into any particular cell type that we are interested in. Importantly, we can also start to use these iPS cells to model disease states. For instance, we can perform genome engineering in these cells and convert them to cells that potentially either recapitulate disease or do not have disease-associated mutations in them, and again, potentially differentiate them into any tissue type of interest. And more importantly, one can potentially complete this loop and enable personalized therapeutics. So with the advent of the human iPSCs, I think our ability to look at human developmental biology has sort of really become powerful. And in fact, most of my PhD work was really focused in the first arm of this loop, where I developed sort of approaches to improve the human iPSC generation process, but also to use this system to understand pluripotency. And towards the end of my PhD, I focused more on trying to look at genome engineering of these iPS cells. So when I went for my postdoctoral work, I continued some of these genome engineering efforts, and towards the end, I also focused on tissue engineering. So I wanted to begin by sort of focusing on my work in the genome engineering aspects. So central to genome engineering is the process of homologous recombination, where if there's a certain region in the genome highlighted here in red, which we wish to modify, then the way we go about doing it is we provide a repair template that is homologous to your target region of interest, but has a corresponding change at the location that you wish to make the change. And if by random chance there happens to be a random cut in the cell in and around the region that you wish to make a change, then the cell can end up reading from this repair template because it is provided in a huge molar excess compared to the sister chromatid. And by the process of homologous recombination, you can make a corresponding change at your target region of interest. But traditionally, the rate of this process is very low and typically only of the order about 1 in a million or 10 million cells get successfully modified using this process. But what was sort of recognized over 2 decades ago is that the key rate-limiting step in this process is the generation of the random cut. And if instead of a random cut, one could make an engineered cut using targeted nucleases, then one can boost the efficiency by about 1,000 to 10,000 fold and bring it into the few percent. And traditionally, these sort of engineered cuts have been made using chimeric proteins. And these chimeric proteins comprise of 2 domains. They comprise of a DNA binding domain and a DNA cleavage domain. And these DNA binding domains have traditionally been programmable DNA binding domains, so this chimeric protein could be recruited to a region of interest. And these have come in basically 2 major flavors. The first are the zinc finger proteins that have been around since 1996. And more recently, we've had a new class of proteins since 2009 called the TAL proteins. Now, both these proteins are heavily programmable, although zinc fingers are less so as compared to TALs. And TALs are again sort of come from bacteria, and they are very large proteins that comprise of these modular repeat domains, each of which can be programmed to bind a specific nucleotide. So while both these approaches, using either zinc fingers or talons, can result in very robust genome engineering, they still come with a range of limitations. The primary among them is that zinc fingers, as I mentioned, are not very programmable, but talons are indeed programmable. But while talons are programmable, they're not the easiest to synthesize. Also, they're very large in size, and they also have this heavily repeat sequence, so it makes it complicated to kind of engineer TALs in a standard lab. And also, neither of these approaches, either zinc fingers or TALs, have really been explored in the context of multiplexability, as in how many multiple changes can you make inside a single cell using either zinc fingers. And in the case of talons, it's hard to imagine that one could do multiplexing just because of the large size of the TALs and the difficulty in being able to deliver these TALs into cells. So both the ease of synthesis and multiplexability are sort of compromised in the case of both zinc fingers and TALs. And importantly, how truly specific are either of these tools remains an open question in many aspects. But more recently, a new sort of tool set has emerged, which is based on the bacterial CRISPR-Cas systems, or the prokaryotic CRISPR-Cas systems. And these are remarkable systems that serve as the adaptive immune defenses of bacteria that use short RNA to direct degradation of foreign nucleic acids. So they come in various varieties. There is type 1, type 2, and type 3. And for the purpose of this talk, I'll focus on the type 2 CRISPR-Cas systems. And just as we imagine adaptive immune defense systems, these systems also are characterized with both an immunization phase and an immunity phase. For instance, in the immunization phase, the system will fortify itself against a foreign pathogen. And this process is not very well understood, but the way this proceeds is that if you look at a typical, sort of an example of a typical type 2 CRISPR locus inside cells, it comprises of these repeat domains adjacent to a cluster of Cas proteins. And now if there's a viral infection, then the virus will inject a certain amount of viral DNA, and a complex of Cas proteins can process this viral DNA molecule, and through a process that is not well understood, incorporate a small piece of this viral DNA into this repeat spacer region, and whereby you have sort of stored a memory of this particular infection. Now, once this memory of this infection is stored, then this system can then serve to protect against a future viral infection. And this is much better understood in large part of the work done right here at Berkeley. And the way it is believed to occur is that this repeat spacer region is transcribed into a pre-CRISPR RNA. And this pre-CRISPR RNA can then associate with a large protein called Cas9 and a small RNA called the tracer RNA. And together they can form a complex on this pre-CRISPR RNA and process it to create this final functional surveillance unit of the CRISPR-Cas system. So this surveillance unit comprises of the large protein Cas9 and the spacer incorporated in the CRISPR RNA and the tracer RNA. And now if there's a repeat infection from the virus, then this surveillance unit can survey the viral DNA molecule. And if there is a match between this spacer sequence and a corresponding sequence in the viral DNA, then it can effect a double-stranded break and result in viral DNA cleavage. So in this fashion, this adaptive immune defense system can both fortify the bacteria against an infection and also use that particular information to fight off a repeat infection. So in terms of being able to engineer this system in human cells, sort of the key observation is that the final functional unit or the main surveillance unit of this CRISPR system is really this Cas9 protein in complex with this couple of small RNA molecules, the CRISPR RNA and the tracer RNA. And so to enable this sort of eukaryotic implementation, really the approach is very simple. In theory, all one needs to do is to express the Cas9 protein with an appropriate nuclear localization signal and express this sort of CRISPR RNA, tracer RNA in a sort of chimeric structure, but the only trick being that you use a polymerase III promoter instead of a standard polymerase II promoter. The idea being that polymerase III promoter transcripts do not get capped or polyadenylated and thus do not get shunted out of the nucleus. So in theory, if one appropriately expresses both the Cas9 and the guide RNA, then one could imagine that they would come together and form this sort of final functional surveillance unit. And before I kind of jump into sort of the details of how this system functions, it's sort of instructive to look at this complex again in sort of higher detail. So we have this large Cas9 protein that can complex with this guide RNA molecule. It really comprises of two major domains. And the domain in green corresponds to the spacer sequence, which is what guides this complex to the target region of interest. And the region in blue is what I refer to as the scaffold region, which is responsible for the association between the Cas9 and the guide RNA molecule. And so once this complex is formed, then it can survey the genome and make a double-stranded break the moment there is a homology between your spacer sequence and a corresponding target in the genome. But the critical aspect is this cleavage event only occurs when there's a small stretch of nucleotides adjacent to the spacer sequence referred to as the PAM that is also present. So in our initial experiments of trying to evaluate how the system works in the human cells, we turn to the process of non-homologous end joining, where if you make a targeted cut at a certain region in the genome, but you do not provide a repair template, then the cells will try to repair themselves, but will often do so in an erroneous fashion and leave behind random deletions and insertions, which can be assayed by sequencing. And indeed, if you make a targeted cut here in the region highlighted in green, and if you assay the population of cells that have been subjected to these Cas9 guide RNA molecules, then you indeed see a range of insertions and deletions at your target region of interest. But so in these sort of early experiments to get a deeper insight into how really this process functions and how truly specific it is, we decided to modify this experiment and instead target two regions in the genome right adjacent to each other, highlighted here in red and green and referred to as the T1 and T2 targets. And indeed, if you had a guide RNA that was targeting the T1 region, then what one found is that most of these insertions and deletions, if you quantified them, were between these two red lines that demarcate the boundary of the T1 target. And now if you expose them to a guide RNA targeting the region T2, then indeed all your sort of insertions and deletions were now confined between the two green lines. So this simple experiment showed that this process is indeed very specific, and indeed you can, in a targeted fashion, either edit the region in red or the region in green, and you could do so in a specific fashion. But importantly, if you exposed the cells to both guide RNAs corresponding to both T1 and T2, then one could make a huge targeted deletion between T1 and T2. And this sort of indicated that the process is also multiplexable, in that one could make multiple changes inside a single cell, and therefore this process was both specific and in some sense multiplexable. And sort of the exciting aspect about the entire CRISPR process was that the workflow was incredibly simple. All one needs to do in theory is now to identify candidate targets in the human genome, which are adjacent to an appropriate PAM site, and then incorporate them into an appropriate guide RNA expression vector. What we also showed in this initial work was that it was possible to sort of synthesize these targets also from DNA arrays and potentially start to create guide RNA libraries. And finally, all one needs to do in this CRISPR system is once you have the appropriate guide RNA expression vector, then to just deliver it to the appropriate cell type of interest. So as a summary of this first part of the work, the tool is indeed robust, and it enables genome editing across a range of cell types, including human iPS cells. It is also highly multiplexable, since really the guide RNAs are only about 100 base pairs in payload, and therefore efficient delivery of multiple of these per cell is readily feasible. And importantly, the ease of targeting of the system greatly exceeds that of zinc fingers and TALs. So since the native system is sort of evolved as a nuclease, one would ideally also want to be able to expand this functionality to other functions. And sort of the next immediate aspects that arose after this initial study was, can we also expand the functionality to include genome regulation in addition to genome editing? And still more importantly, really how specific is this system? Because if you're going to use this in a targeted editing fashion, then really we need to know what is the landscape of specificity of this CRISPR system. To expand sort of genome regulation, the key aspect to do is to basically take the original Cas9 guide RNA complex and make a nuclease null version of the Cas9 protein and hope that it still it still binds to the guide RNA and can do RNA-guided genome targeting. And this was feasible because one could align the protein against existing sequences in the database, and we were able to putatively identify 4 nucleotides that were, 4 positions that were responsible for magnesium coordination and create a quadruple mutant of the Cas9 that showed no detectable nuclease activity as compared to the wild-type Cas9 protein. So one could make a nuclease null version of the Cas9, and which showed no activity. But to sort of show that it also retains RNA-guided DNA targeting ability, we then next tethered the Cas9 protein with a transactivator domain, referred to here as the Vp64 activation domain. And to assay if this still functions as an RNA-guided tool, we looked at 2 reporter constructs inside human cells. And the way these reporter constructs work is there's a minimum promoter that drives the fluorescent protein of interest, but upstream of the minimum promoter is a guide RNA binding site. And this guide RNA binding site is unique between the 2 reporters. And upstream of the guide RNA binding site is a shared TAL target binding site that is shared between both the 2 reporters. So indeed, if we excited these reporters using a TAL transcription factor, then one could turn on both the reporters effectively. But if we use just the nuclease null version of the Cas9, then neither of the reporters was effectively turned on. But if you use the appropriate guide RNA corresponding to the appropriate reporter, then the corresponding reporter was turned on. For instance, here guide RNA1 turns on reporter 1 and not reporter 2, and vice versa, where you have guide RNA2 that turns on reporter 2 and not reporter 1. So this indicated that the nuclease null version of the Cas9 is indeed functional, and it can bind to guide RNAs and also do RNA-guided targeting. And in particular here, by attaching an appropriate effector domain, you could convert the Cas9 system into a transcriptional activator. So while this worked in the context of reporter constructs, we wanted to also see if this would work in the context of endogenous human genes. And in these early experiments, I decided to focus mostly on genes that were associated with pluripotency. In particular, in this experiment, we focused on the REX1 gene. And since we didn't know how exactly one would go about turning on the REX1 gene, we decided to examine a length of sequence about 5 kb upstream of the transcriptional start site. And we designed about 10 guide RNAs that were located at various positions along this stretch of DNA. But what was very interesting when we repeated these transcriptional activation experiments is that individual guides were not stimulative in transcription. And most of them just resulted in about a few-fold activation of transcription at this endogenous locus. It was only when multiple guides were sort of combined did we have sort of any measurable transcriptional activation from this endogenous locus. And typically, in our experiments, it took at least 4 to 5 guides to significantly turn on an endogenous gene. So for endogenous genes, the sort of key conclusion was that single guides were not effective, but this heavy synergy between multiple sort of Cas9 guide RNA complexes was critical for transcription regulation. And this was also true in the context of 2 other genes that we examined, the SOX2 gene and the NANOG gene. And in the case of one, we could activate the endogenous gene by about 80-fold, and another in the case, in another by about 1,200-fold. But once again, it was critical that only once we had 5 to 10 guide RNAs that were used simultaneously was any sort of robust transcriptional activation observed at any endogenous locus. So in sort of summary for this part of the experiment, nucleus null Cas9s tethered to effector domains do retain RNA-guided genome targeting functionality, but RNA-guided gene activation is not very robust, but requires synergy between multiple Cas9 guide RNAs for effective transcription. So this actually remains a very active area of research interest for me as to why the system is not very robust in the context of genome regulation, and what one can do to sort of improve this genome targeting in this sort of genome regulation functionality of the Cas9 system. But what this ability to be able to do transcriptional regulation using the Cas9 system enabled was it enabled us to look at the question of specificity. In particular, one could now start to design next-generation sequencing-based assays to basically look at the landscape of specificity of DNA-binding proteins in general. And the approach that we devised for this particular approach was, can we use transcriptional activation to look at the specificity of any DNA-binding protein of interest? And again, sort of looking at the reporter that I had described earlier, what we did was instead of using a single reporter, we made a library of reporters, but with the key aspect that in front of the minimum promoter, which drives your fluorescent protein of interest, we also inserted about a 24 base pair barcode that was unique among every element of the library. And upstream of the minimum promoter is your corresponding target site that you wish to interrogate, but we also made a library of mutants corresponding to the target site. So there was a library of reporters where every particular mutation in the target site was associated with a unique barcode in your fluorescent protein of interest. So now if we stimulated the library with a transcription factor that bound a site that was shared between all the library elements, then one would expect that it would turn on all the corresponding library elements uniformly. And that was indeed true when we used the control transcription factor. Then sort of the distribution of the barcodes that we observed was similar to the theoretical distribution of barcodes that one would expect for a library of interest. But now if we use the transcription factor that bound the corresponding target site that we wanted to interrogate for in terms of specificity, then the target transcription factor would turn on those particular elements strongly that it preferred to bind still more, and it would thus bias sort of the expression of the barcodes accordingly. And indeed, what one finds is if you use a TAL transcription factor, for instance, in this example, binding an 18-mer site, then it will skew the distribution away from the theoretical distribution of the barcodes. And if one took a ratio between the TAL transcription, the expression from the TAL transcription factors versus the control transcription factors, then one would get a specificity landscape. And in particular, in this particular example, if you take an 18-mer TAL transcription factor, if there are 0 mutations in the target site, then one indeed observes a strong stimulation. But the moment there are 1 or 2 mutations in your target site, then your expression very rapidly falls off. And that is what one would expect from a specific protein, where most of your activity is against your wild-type target. But as you have mutations in your target, then your specificity should fall off. But what is remarkable is that this is not a step function in many ways, but is what one would like for a specific protein. But there is this sort of tolerance for multiple mutations in your target site. But what is the case in the case of, but what is really the scenario in the case of the Cas9 guide RNA system? For instance, here is an example of a Cas9 guide RNA system that has been evaluated in the context of this library. And indeed, what one finds is if there are 0 mutations in your target site, then you have high activity. But the moment you have a single mutation, your activity drops down very remarkably. And with multiple mutations, your activity diminishes very significantly. And one can now go in and look at your library at greater detail and start to examine sort of single base pair mismatch data. And what one starts to observe very quickly is that there's a very interesting aspect about the specificity, in that most of the specificity comes from the 3' end of your guide RNA, where any sort of mutations in your target region at the on the 3' end result in a rapid loss of activity, but any mutations on the 5' end typically do not result in loss of activity. And this is also true in the case of the double mismatch data, where consistent with before, if you had multiple mismatches, in this case 2 mismatches, then your activity as with respect to your wild type falls down dramatically. And one basically observes that the system is very specific in the context of this guide RNA. But what we also observed is that while this particular guide RNA was very specific, one could now examine a second guide RNA which was not very specific. For instance, in this guide RNA, even with 1 or 2 mutations in your target site, one still retains a large amount of activity. And again, if you examine the single base mismatch data, then while the former guide RNA was specific in the 3' region of the target, the second guide RNA is not very specific on the 3' end of the target. And this is, again, recapitulated in the 2 base mismatch data, where the first guide RNA completely loses activity upon 2 mismatches. The second guide RNA still retains activity upon mismatches in the 5' end of your target region. So in terms of summary here, the Cas9 guide RNA complexes can, in general, tolerate about 1 to 3 mismatches in their target DNA sequence, which is not too bad in many ways, because one could then appropriately bioinformatically choose guide RNAs that were greater than 3 base mismatches away from the genome. But the important aspect was that this specificity profile was very highly guide RNA dependent, and it remains to be identified as to what guide RNAs are the most specific and what sort of nucleotide patterns affect specificity. The other aspect that we also discovered from these experiments was in the context of the pyogenies. The PAM was both NAG and NGG. And so clearly sort of the important question that comes to mind is, well, if you can tolerate 1 to 3 base pair mismatches, then how can one start to improve the specificity of the Cas9 guide RNA system? And really there are 2 major approaches that one can take. The first is, of course, can we control the duration and dose of the Cas9 guide RNA expression? And clearly it's a kinetic argument where if you have sufficient duration and dose of your Cas9 guide RNAs, then one would eventually hit almost all your off targets. So providing an appropriate pulse of Cas9 guide RNA expression, typically, I think, in my opinion, the best approach is to use RNAs, then one can have very high specificity. But the other approach is similar to the zinc fingers and TALs. Can we also engineer a requirement for cooperativity? For instance, with zinc fingers, you need 2 zinc finger proteins to come together and make a double-stranded break. And similarly, for the TAL proteins, you require this dimerization of FOC1s, and thus 2 TAL proteins to come together and make a break. So can we do a similar thing in the case of the Cas9 system, where can we actually engineer a requirement for cooperativity? And indeed, the one approach for doing so is the one that we explored, was this concept of using offset nicks to create double-stranded breaks. And here the idea is that if you take the Cas9 protein, which makes a double-stranded break by making 2 nicks on opposite strands of the DNA, and make a nicking version of the Cas9 protein, then one would then require 2 Cas9 proteins to come together, Cas9 guide RNA complexes to come together in a sort of coordinate fashion, and only when nicks were created on opposite strands would one have a double-stranded break created. And that was sort of the offset nicking approach. And key to the offset nicking approach to work is the aspect that single nicks typically do not result in any sort of scar activity in cells. So typically cells are very good at repairing single nicks. And if an off-target single nick event happened, then cells would end up repairing them very efficiently, and one would not observe any sort of NHEJ event. So only when 2 nicking events happened in a coordinate fashion and on opposite strands of the DNA, then one would observe a double-stranded break, which would be measured as an NHEJ scar. And so how does the system work in the context of an endogenous locus? For instance, here we have 8 guide RNAs targeting a particular region in the genome, specifically the AVS1 locus. And if you use just the wild-type Cas9 protein, then one can see that each of these guide RNAs results in very robust activity from about 20 to 70% targeting. But if you use just the nicking version of the Cas9, then as expected, none of these guides can result in any sort of scar. But when we start to use these Cas9s in a coordinate fashion and generate either 3' overhangs or 5' overhangs, then one starts to observe robust NHEJ events. In particular, what we observe very interestingly is that only when there are 5' overhangs that are generated is that one observes very robust non-homologous end joining. And that is kind of expected in the sense that if you look at the conventional homologous recombination pathway, then it is the 3' end of the DNA that invades your donor strand. So one would assume that sort of, if there's a 3' overhang that is generated, then one would not observe robust NHEJ. But if you have 5' overhangs, then one can imagine the polymerase filling it up and the 2 strands not being able to come together and repairing themselves effectively, and one would observe robust NHEJ. So indeed, one observes robust NHEJ when you have this sort of cooperativity between 2 Cas9 guide RNA molecules. So in summary, improving efficacious targeting using the CRISPR-Cas systems entails either appropriate bioinformatic evaluation of candidate targets or engineering a requirement for synergy in effector functions. So the Cas9 system can be used for both RNA-guided genome editing and regulation. It's sort of the major conclusion from this sort of work, and one has strategies to sort of improve the specificity of the system by employing a synergy in effector functions. But while this Cas9 guide RNA system can be used for both genome editing and regulation, for engineering complex systems, one needs to have this ability to perform multiple effector functions simultaneously. For instance, while one can take this Bioengineering Cas9 guide RNA system and convert it to an activator, for instance, and potentially also a repressor, but if you wanted to perform activation and repression simultaneously inside the same cell, then it would not be feasible because the activator could bind to the repressor guide RNAs and the repressor could bind to the activator guide RNAs, and therefore it would not be possible to have multiplex functionality inside the same cell. And this is unlike in the case of TALs and zinc fingers, where they are just passive DNA binding proteins, and each of them can be given their own effector function. So sort of to solve this last aspect of the genome engineering toolset, we decided to engineer orthogonal Cas9 guide RNA systems, where each Cas9 guide RNA system could have its own effector functionality, and they would not, and these Cas9 guide RNA systems would not interact with each other. And sort of the motivation behind the study, or the idea behind the study was, can we actually find orthogonal Cas9 orthologs? And there are at least 2,000 Cas9s that have been documented from metagenomic literature. And the idea was, are there orthologs present in this sort of database that are orthogonal to each other? So how does one go about finding orthogonal Cas9 orthologs? So the idea was, if you look at the Cas9 guide RNA complex, then the ability of being able to form this complex is in large part determined by the sequence in this hairpin, which is identified in red. So could one then look in literature and then find Cas9s that were highly divergent in this sort of sequence? And so that was our approach to identifying orthogonal Cas9 orthologs. And indeed, we found 3, one from Pyogenes, Thermophilus, and meningitis, that had very divergent sequences in this hairpin loop. And then the hypothesis was that if we took these 3 Cas9s and then we exposed them to the appropriate guide RNAs, then they would only bind to their corresponding guide RNAs. And in this fashion, each of these could be given their own novel effector functionality, and then one would have orthogonal Cas9 guide RNA systems. And so how orthogonal are these in an experimental setting? They are indeed, sorry. Okay, it's all kind of hung. Sorry about that. Hey, it's kind of frozen here. Okay, sorry about that. I don't know what to do. Oops.
Can I ask you a question?
Sure, go ahead.
So you're delivering endonuclease. How does that exactly affect transcriptional activation? Are you removing a repressor element? Or are you actually delivering a transcription factor that activates specific transcription with the guide RNA?
So we create a nuclease null version of the Cas9, and that nuclease null version of the Cas9 is then tethered to an appropriate effector domain.
So you're basically just bringing some complex that's not necessarily an endogenous transcription factor that, upon binding, structurally opens it up and allows for transcription?
No, so we're using a very standard activation domain called the Vp64 activation domain that comes from a viral DNA, from a viral protein, and that has been known to be a fairly universal transcriptional regulator in many ways.
But you're delivering that with the guide RNA?
Yes.
As opposed to
Yes, yes, that's true. Sorry, I can take more questions. Sorry, I don't know what is going on here.
That's great. You get on track. Get on track.
Okay. It's just totally frozen. Sorry about that. Yeah. I do have it on Dropbox and thumb drive. Sure. Sure. Okay. Okay, I'm just going to quickly restart. Hopefully that should solve the problem. I think it should be okay. Oh, no, I think it should work. It should work. I mean, it's the first time it's doing this, but all right. Okay. All right. So sorry about that. So the idea was, can we have multiple Cas9 orthologs which have divergent CRISPR sequences that they bind, and then could they indeed be actually orthogonal in an experimental setting? And so how orthogonal are they in an experimental setting? So for instance, here you have 3 different Cas9s, the SP, ST1, and NM, as I had identified earlier. And what one indeed finds is that if you take the SP Cas9, then it only has robust targeting in the presence of its corresponding guide RNA and not in the presence of any of the other guide RNAs. And indeed, that is similarly true for the ST1 Cas9. That shows its most robust activity in the presence of its guide RNA and not any of the other guide RNAs, and so on and so forth. So indeed, these 3 Cas9s are very orthogonal to each other, and in theory can be used to attach appropriate effector domains onto each of these Cas9s and do orthogonal sort of genome regulation functionalities. So in summary, the orthogonal Cas9 guide RNA systems, they enable both individual and simultaneous effector functionalities. But they also help us establish a sort of comprehensive first-generation Cas9 genome engineering toolset. And the way I'm trying to look at the Cas9 system is that the Cas9 is, in some sense, a unifactor. It's the only known example in biology where one can programmably bring together RNA, DNA, and protein. And in that fashion, the Cas9 system can really be used to engineer a range of functionalities. For instance, you can use a nuclease version of the Cas9 and use it to make cuts and deletions. The nickase version of the Cas9 can be used to make nicks or offset nicks, as I showed. One could use the Cas9 nuclease null versions and attach appropriate effector domains onto the Cas9 protein and use it for either genome regulation or attach novel effector domains such as fluorescent proteins or monomeric nucleases, or in my opinion, probably one of the highest value things would be to convert it into a recombinase. And similarly, one could take the nuclease null version of the Cas9, and instead of attaching effectors onto the Cas9, one could attach effectors onto the guide RNA by using aptamer domains and using those to recruit aptamer binding proteins and use it for genome regulation. And I didn't get a chance to show some of this data here, but one can do this effectively. And one can also instead use this system to bring together RNA, DNA, and protein in a sort of very complex fashion and start to study processes such as transcriptional regulation in a way that has not been examined before. So the way I look at the Cas9 system is that, in some sense, it's a unifactor and at least has the potential to do a range of functions that go beyond just plain genome editing or genome regulation. So in sort of in this quest of moving from reading genomes to interpreting genomes, I wanted to switch to a second project that I spent almost an equal amount of time working on, which is on the fact that while systems such as the Cas9 system can enable us to make multiple changes inside cells, we also need systems that enable us to have this sort of multiplex readout from cells. So can we make network level perturbations, but can we also have network level readout? So if we make multiple perturbations in a network, then we want to be able to also assay the consequences of what happened with this perturbation. So can we make new tools in the direction of being able to do very heavily multiplex in situ monitoring? And traditionally, this has been done in the context of fluorescent proteins, but they are limited in multiplexability. So we ideally want a new system that is still compatible with a standard 3-color microscope, but can be much more multiplexable and be used for in a sort of live cell barcoding scheme. And in this direction, sort of the solution again came from DNA binding proteins in many ways. And the idea was, can you actually use programmable DNA binding proteins as barcodes? And more specifically, if you look at the programmable DNA binding protein and the DNA as a receptor ligand pair, then it's a remarkable system in biology where both the receptor and the ligand are programmable. Both the DNA binding protein is programmable and the DNA is programmable. So really the space of engineerable orthogonal interactions is really huge. And clearly one can use this programmability for engineering macromolecular associations that go beyond genome engineering. In particular, the idea here was, can we actually take programmable DNA binding proteins, for instance here the zinc finger protein, and instead of using it in the context of the nucleus, can one actually express it on the cell surface? And if one can express a zinc finger protein on the cell surface, then one can expose the cells to an appropriate double-stranded DNA molecule that is the ligand for this particular zinc finger protein and use it for labeling purposes. So how does the system work in a live cell setting? For instance, here there are 2 cell types that are mixed together, one expressing zinc finger 1 and another expressing zinc finger 2. And they have been exposed to a fluorescent DNA ligand. And here is a scan, a confocal scan of the mixture of these 2 cells. And indeed, what one finds is there are these sort of beautiful ring-like structures that form around the cells because these are cell surface proteins that have been exposed to this fluorescent ligand. And if one just zeroes in on the signal from the zinc finger 2 probe, then that is kind of shown in red here. But if one overlays the signal from the zinc finger 1 probe, then what one finds is that one can indeed do very multiplex labeling in a highly cell type specific fashion. But then how does one go from standard 2 colors or 3 colors to more than 3 colors? And so the idea here is that one still uses 3 colors, but one uses these 3 colors in a sequential fashion. The idea being is now if you have cells expressing the zinc finger protein on the surface, then you expose them to an appropriate DNA binding probe. And this probe really has two components in it. The first is a DNA binding, the double-stranded DNA domain, which is what the zinc finger binds to. But then what one does in the first step is one exposes the probe to only one fluorophore. So for instance, here there are 6 regions which have 6 different zinc fingers being expressed from a population of cells. But in the first step, one just exposes this probes corresponding to 3 of the samples, here given by green, red, and blue. But in this next step, what one does is one extinguishes the signal from the first set of 3 probes. And in this fashion, now one can, in the next step, expose the remaining three samples to a fresh set of fluorophores. And indeed, what one can do now is extinguish a signal from the first three samples, and now visualize the next 3 samples, again using the 3 colors as green, red, and blue. So in proceeding this fashion continuously, one can now start to probe living cells with, at least in theory, an infinite number of colors. But the critical thing of also having zinc fingers on the cell surface is that they are also versatile handles for performing other functions. For instance, one can use these cell surface zinc fingers for cell capture. For instance, here you have a mixture of cells. And in theory, if one of them expressed the appropriate zinc finger domain on the surface, then one could use magnetic beads that have a double-stranded DNA conjugated on them and use them to selectively capture a particular cell type from a mixture. And indeed, that is feasible. For instance, here you have 2 cell types mixed together, and you can selectively enrich for the one that expresses the corresponding zinc finger by using magnetic beads that pull out that particular corresponding element in the mixture. Importantly, one can go beyond cell capture, and one can even start to do targeted delivery. For instance, one can take lentiviruses, which are very easy to pseudotype to an appropriate, to basically pseudotype to bind appropriate receptors, and one can now start to make pseudotype lentiviruses that are actually pseudotyped with double-stranded DNA molecules. And indeed, now if you take these, if you take just a plain lentivirus that does not have the appropriate double-stranded DNA molecule present on them and expose them to zinc finger expressing cells, they do not infect the cells very readily. But if you have the appropriate double-stranded DNA probe conjugated on them, then one can do effective delivery inside cells. So one can also use these handles for targeted delivery. And still more importantly, one can start to use this to engineer cell-cell interactions and cell-substrate interactions in a programmable fashion. For instance, one can take oligoconjugated cells and expose them to oligoarrays and start to pattern cells in complex patterns, for instance, here a checkerboard fashion. So using cell surface zinc fingers as barcodes, they enables multiplex labeling and imaging of live cells. And it also provides a versatile cell surface handle for applications in cell and tissue engineering. So in terms of, so I just wanted to conclude by saying that in terms of sort of reading genomes to interpreting genomes, I think we are at a very interesting phase in biology where there is this confluence of very rapidly maturing tools. And this is one of those unique phases where there are tools now that we can use to recapitulate biology, for instance, use induced pluripotent stem cells. We have tools to perturb biology, such as zinc fingers, TALs, and CRISPRs. And importantly, our ability to also assay biology is rapidly growing as our tools for imaging, sequencing, proteomics, and metabolomics really becomes very powerful. So really, in that sense, it's an interesting phase in biology where there is this sort of rapid confluence of these emerging tool sets, which are getting very rapidly much better as every passing day goes on. So in terms of the particular biological problem that has fascinated me, where I want to apply these sort of assay of tools is the context of development. For instance, what fascinated me first in graduate school was this aspect that a single totipotent zygote can give rise to this multicellular organism. But the key aspect from an engineering standpoint is that it does this sort of very complex process in a very reproducible fashion, so much so that we can start to imagine, one can sort of very effectively draw CAD diagrams for organs. But then how do we go from a CAD diagram of an organ to sort of a transition and sort of make this sort of, take a CAD diagram and actually convert a sort of living tissue out of it? And traditionally, the way one can look at this process, if you take a very sort of reductionist approach to this process, there are sort of 2 ways of looking at it. One is what nature does, which is innate self-assembly. And from a biomedical engineering perspective, we have the sort of engineered assembly approach using biomaterials, or like in the case of, say, cell surface zinc fingers, can we actually control cell-cell interactions? But then can we actually bridge these 2 aspects of innate self-assembly and engineered assembly? And that is sort of the broad research direction that I kind of want to pursue in the future. And that is indeed, I think, feasible because with the advent of the Cas9 system, and especially the ability to make these guide RNA libraries, one can start to use the human iPSC system as a model, and not only start to map developmental gene networks and identify what are the key developmental gene networks, but one can also now start to program these and control and make sort of programmable differentiation networks and sort of combine them with programmable biomaterials and start to engineer organogenesis. So that is sort of the broad research direction that I wanted to explore moving forward. And I just wanted to conclude by thanking the people on this that were responsible for this study. George Church, who was my mentor for the past 3 years and who's also very kind and an amazing mentor for me. And the key people who were responsible for the study, John Uck, Luhan Yang, Kevin Eswelt, and Mark Boosburner. Also members of the Church Lab, and Dave Zhang, who was a key collaborator on the cell surface zinc finger project. So thanks, and I can take questions now. All right.
I have a question about the Cas9. Is that able to bind silence DNA, so those wraparound histones, et cetera? Can that machinery still find those regions of DNA, or is it only as efficient as you've shown it on the active regions?
I think that's a good question in the sense I don't think anybody has systematically examined that aspect carefully. But what I do find is that so-called repressed loci are targetable by the nuclease. Like you can go into a repressed locus and make a double-stranded break. The downstream assay of homologous recombination at a repressed locus is much harder. So I'm able to make a targeted cut at a repressed. But then was there an exact histone position at that point where I made the cut? I never assayed that very carefully. But in terms of being able to target repressed loci, I think it's feasible, just from the perspective of looking at the cut. But now if one turns it around in the context of genome regulation, what I typically find is that if you want to do genome regulation at an endogenous locus, genes that have at least a basal level of expression by qPCR with respect to your appropriate controls, those are the most stimulative, in the sense you can stimulate them by 500 and even 1,000-fold. Genes that are almost completely repressed are much harder to turn on using the genome regulation system. But now, again, genome regulation is so complex that there could be many factors that impinge on this. So there are these so clearly the repressed regions are hard to target, but they are targetable with the Cas9 system. I mean, at least one can make a double-stranded break, but the HR is the complex sort of process there. But regulation is kind of indicating that it's probably harder from another perspective.
Have you tried recruiting cell lasers?
So I think one of the keys to actually improving the Cas9 guide RNA system is actually going to be to not just rely on a transcriptional activation domain, such as the Vj64 domain. I mean, although it's a powerful domain and a fairly universal domain in being able to recruit the polymerase II promoter, but clearly we have to look at sort of epigenetic modifiers. And perhaps I think maybe look at other Cas9 orthologs, because one of the things that kind of emerged from that ortholog, the Cas9 ortholog story, was that not all orthologs were sort of created equal. Some were better than others, and some were quite terrible in sort of activity, for instance.
So there is another, ramification of this IPS technology, which is direct transdifferentiation, for example, from
Sure.
fibroblast to neuron,
Sure.
fibroblast to plasma monocytes, which usually people just overexpress like 3 or 5 transcriptional factors.
Sure.
Do you think CRISPR system will be better in induction of endogenous set of factors, or even providing a control system? As compared to what people already shown?
I think the CRISPR systems can have a very unique advantage in that system, in that paradigm. So like, clearly we know a few transcription factors that are important in that process, and one can use that to make the transdifferentiation process. But where the CRISPR system can contribute is on 2 fronts, I feel. First is in the discovery of these in a rational fashion. Currently, we kind of mine literature and see what are sort of the critical factors out there, but can we actually identify them in a sort of unbiased fashion, number one. And I think the second aspect where it can make a big difference is, while it's possible to overexpress a few transcription factors, I mean, there are a lot of proteins that are very large, very hard to deliver. Probably in the case of non-coding RNAs, I mean, they can be over 10 kb in size, and so if they are critical for a certain biological function, then pretty much the only way one could perturb those would be in the context of using a CRISPR genome regulator, for instance. So I feel on those 2 fronts, both sort of discovery as well as being able to do some very complex cis regulatory functions would be where the Cas9 system would really trump existing approaches.
And probably also screening how neuronal those cells are. Are they really neurons? Are they really cardiomyocytes or something in between?
Sure.
So can you incorporate like a multiplex reporter system?
Sure. Yes. I think reporters is going to be a very, very valuable resource that one can generate using the system.
The data that I thought was interesting was the guide RNA and the relative expression. And at one mutation, you had like the greatest variance in that box plot.
Right.
Can you explain that?
There's something we're trying to delve in deeper and see. Like, even if you have 1 mismatches, some positions you can have still high activity. Part of the reason for the variance was also because of the fact that I used this library-based approach, and I did not. So part of the reason was that we did not have enough deep sequencing data. data for those experiments. So if we had sort of more coverage for our libraries, then some of the noise would have gone away. So part of the noise was from just lower coverage in the next generation sequencing experiments. But part of the noise also comes from the fact that certain mismatches are almost completely well tolerated. So, but we haven't sort of delved deep enough to kind of, you know, get a sort of a deeper insight into why some are better tolerated than others.
So when you showed that multiple guide RNAs binding or expressing multiple guide RNAs for the same gene would increase the transcriptional activation by a lot as compared to just one.
Right.
So is that more because there's a higher probability that one of them will bind, or is it because when those multiple regulatory factors are bound simultaneously, there's more like opening of the DNA, more transcriptional activator recruitment?
So I believe it's actually the latter. So there's clearly a synergy is what one observes. So it's greater than the sum of the parts. So if you have 5 together, then the activity from those individual ones, if you take the summation of them, that's significantly less than the 5 together.
Could you similarly just recruit a lot of helicases using the same Cas9 or like scaffold a bunch of activation domains
Oh,
together?
absolutely. I mean, I think really, in fact, there's a lot of sort of emergence in biology recently that clearly there are, like these super enhancers, for instance, where you really have this really, like hundreds and hundreds of transcription factors that are really coming together at a locus to really boost transcription by several thousand-fold. So clearly, I think this aspect of really having a heavy amount of synergy at a transcription locus is going to be critical to have really robust sort of turning on of an endogenous locus. Though we do find that a few single guides can give some measurable activity, but in general, it's much harder. It's much harder.
Is there any conceivable thought of using this kind of targeting to bring like chromatin modifying complexes like HATs or methyltransferases to essentially play with selected epigenetic modification?
Absolutely. I mean, I think it's like there are several groups that are trying to explore that particular research direction for sure. I mean, I personally have not looked at that aspect in greater detail. I've been looking more on the sort of specificity side of things recently. But definitely, I mean, that's one of the aspects that can be very easily done. Like one could recruit methylases, demethylases, acetalases, deacetalases. I mean, you have this whole range of effector domains that one could recruit out there, for sure.
So in your presentation, you switched from Cas9 to zinc nucleases at
Right.
the end. So you said it is used for cell partitioning and also target payload. So you mentioned with the very beginning, Cas9 has its own advantages. Then why did you switch to zinc nucleases?
Oh, why I switch to zinc? So the thing is, if you want to express something on the cell surface, the Cas9 guide RNA complex doesn't make sense because you have your specificity would come from the guide RNA in many ways, right? So but in the case of zinc fingers and TALs, what I found was I explored both zinc fingers and TALs, but only the zinc fingers were more robustly put on the cell surface. So TALs were very difficult to put onto the cell surface using a range of transmembrane domains. So zinc fingers, just because of their very compact size, were very easy to sort of put onto the cell surface in that sense. So, and I do tend to believe that each of these DNA binding domains will have its own niche application. And I think here's one example where zinc fingers are very potent as compared to others, for instance. And I think even in the context of endogenous targeting, I mean, I would not rule out any of these technologies for now, though of course the CRISPR system is just wonderful for its ease of use in many ways.
Last question.
Right.
So when you use the guide RNA and Cas9 system to targeting, is the location or efficiency of the targeting affected by the physical organizational property?
It is to some extent, yes. So I mean, definitely there are regions of the genome that are easier to target versus the other, but what I do find, and as I was mentioning to you, is repressed loci can be targeted using the Cas9 guide RNA system. It's just that like for instance, homologous recombination, which is really a two-step process, those get affected more dramatically. But that's sort of an observation based on a few guides so far. It probably has to be scaled up to a few thousand to make it sort of a more solid argument. All right, thanks.